Hello, everyone. Welcome to the session of Sexual Reproduction in Flowering Plants, Theory Part 6. Disclaimer: the images, text, and other material used in our presentations are for educational purposes only. We respect the intellectual property rights of the original creators and owners. If you are the owner and have any concerns or issues, please contact us at sathi@iitk.ac.in and we shall remove the content immediately. Please note that the part of the content may have been generated using generative AI, which is experimental. So, this is the learning objective of today's lecture. To understand the stages of embryo development in monocot and dicot and also endosperm development. So we are going to study about the development of the embryo, the development of the endosperm in the case of monocot as well as in the case of dicot plants. So this is the learning objective. Now, today's index: post fertilization structure and events. This whole topic, like embryo and endosperm development, comes under the post fertilization structure and events 1.4 section of your NCRT. And in that, the subsection we are having the endosperm 1.4.1 and embryo 1.4.2. At the end, we will summarize today's lecture. So let us start today's lecture with post-fertilization structure and events. So following double fertilization, events of endosperm and embryo development, maturation of the ovules into the seeds and the ovary into fruits are collectively termed as post-fertilization events. So when you see any flower that got fertilized, what are the changes you see externally? What you observe those changes when you see any flower got fertilized? Anyone? What type of changes you see in a flower when it got fertilized? Ovary become fruit. Yes, ovary becomes fruit. Any other? Ovary becomes fruit. And ovule changes to seed. Yes, this is the second change we see. But there are some other changes also, like the flower wither away. The flower wither away. All the floral parts like sepals, petals, androecium, they fall off. Okay. Yes, petals and sepals fall off. Androecium also fall off. Okay. But the pistil part, the style and stigma part dry up, but the ovary swells up. The ovary swells up and forms the fruit. And inside the fruit, what is there? Inside the fruit, there is ovules. Inside the fruit, we are having ovules, and after fertilization, this ovules changes to seed. Ovules changes to seed, and ovary into fruit. So this change comes under the post fertilization events. These all changes which we have discussed just now is the post fertilization event. But there are two more events. There are two more events that, okay, the thalamus part. What happens to the thalamus? Anyone can tell me what happens to the thalamus? Hmm? The thalamus squeeze there. It gets squeezed because when you see any fruit, did you see any thalamus except the apple or the false fruit? What about mango? What is the mango we see there? The, the fruit part. That is the ovary. But do you see any thalamus there? So the thalamus becomes squeezed there. Okay. And you cannot see any bulbous part or a flat part through which the ovary is attached. Okay. Now, after the double fertilization, the two other changes that occur, inside the ovule, that is development of endosperm, development of endosperm, and after the development of embryo and endosperm. These two changes are also there. There is a development of endosperm and embryo. So, what are the changes? Formation of embryo, formation of endosperm, whole ovule changes to seed, ovary changes to fruit, and all the other parts they got wither away. Okay, okay. So, let us see endosperm first. Now, there is one point: which part develops first, endosperm or embryo? Which part develops first, endosperm or embryo? And if endosperm, then why? Why the endosperm develops earlier than the embryo? For nourishing, because the embryo development required nourishment, and that nourishment is supplied by the endosperm. Correct? Okay, that nourishment is supplied by the endosperm. So we will see how the endosperm formation takes place. First thing, so endosperm development precedes embryo development. After the development of endosperm, there is a development of embryo. So this is very important point. And the question is why? The primary endosperm cells divide repeatedly and forms a triploid endosperm tissue. They form the triploid endosperm tissue. And this endosperm tissue provide the nourishment to the developing embryo. That provide nourishment to the developing embryo. The cells of this tissue are filled with the reserve food materials and are used for the nutrition of the developing embryo. So here, this is the question, the answer of the question why, right? The cells of this tissue means the endosperm, it is filled with the reserve food material. It is having lot of reserve food material and that are used for the nutrition of the developing embryo. Can anyone can tell me why is the reserve food fructose? Actually, the food is transported in the form of fructose, okay, and it gets stored there in the form of fructose. But later on, when they want to use up, it convert them into the glucose only, right? Yes, fructose. Now, I want to ask a question here that the reserve food material is provided by the endosperm only. Okay, why not the other tissues are involved in this? Why this endosperm is more concerned for the providing nourishment? The answer is this, that the ploidy of the endosperm is 3n. The ploidy of the endosperm is 3n, triploid. So whatever the food material is synthesized, the protein, okay, the different secretions, they are having triploid nucleus. It means 3 pairs of the chromosomes. So they can synthesize more number of protein and that is required for nourishing the developing embryo. Okay. It's not only the fructose, that could be like here, the proteins are more important. Okay, so there are different enzymes, different proteins that are required for the developing embryo. So all that are provided by this endosperm. In most, in the most common type of endosperm development, the PEN means primary endosperm nucleus undergoes successive nuclear division to give rise to the free nuclei. So here, what does this mean? Free nuclear division. Hmm? Free nuclear division. The nucleus is repeatedly dividing 1 to 2, 2 to 4, 4 to 8, like this. Okay, it is repeatedly dividing, but the cell wall is not laid. The cell wall is not laid, or you can say the cytokinesis is not there. The cytokinesis is not there. So in most common type of endosperm, no, here the cytokinesis simultaneously it is not taking place. Simultaneously it is not taking place. Actually, there are the three different types of endosperm. Three different types of endosperm. But in your syllabus, it is not mentioned that. It is only mentioned the nuclear endosperm. There are three different types of endosperm: nuclear endosperm, cellular endosperm, and halobial type of endosperm. I'll tell you what are these, but in your NCRT, it is only mentioned the nuclear, okay, uh, nuclear endosperm. So, in what happened in the case of nuclear endosperm? The nucleus is continuously dividing without any cytokinesis. Okay, so there are thousands of nuclei. There are thousands of nuclei, right? So this primary endosperm nucleus continuously dividing and dividing, and that is why it is called as successive nuclear division. Successive nuclear division. The nucleus is dividing continuously, and there are number of free nucleus. Number of free nucleus. This stage of endosperm development is called as free nuclear endosperm. Okay. So this stage of endosperm development is called as free nuclear endosperm. No cell wall is laid till here. Now what happened after that? Let us see that. Subsequently, the cell wall formation occur and the endosperm becomes cellular. But once the number of nucleus has been formed, slowly and slowly the cell wall is laid around each nuclei. Okay, later on the cell wall is laid along each nuclei, and that is forming a cell. So that is a formation of cells in the endosperm. Okay. Now you see coconut, and that too green one. That too green one. What you consume from those green coconuts? Coconut water, right? You consume coconut water, you drink that, and it is very healthy also. So when you drink, actually you are drinking that endosperm, which is full of nuclei, So the number of free nuclei formed before cellularization varies greatly from endosperm to endosperm, means in the different species of the endosperm. The number of nuclei is not fixed, it varies. Okay. Now the coconut water from the tender coconut, the one you consume for the drinking water, right? So that you are familiar with is nothing the free nuclear endosperm made up of thousands of nuclei, and the surrounding white kernel is the cellular endosperm. So this liquid nucleus, the liquid part which is having lot of nucleus, is slowly and slowly settled, and the cell wall formation takes place, and it becomes the cellular endosperm, right? And when you see the mature, yes, the kernel, that is the malai, that vendor sells, give you the malai of that coconut, and that is the cellular endosperm part. That is the cellular endosperm part. Endosperm part. Okay. And what about the brown one, that the hard one, coconut? All the water is, all the water is consumed up. All the nucleus is laid there, layer by layer, and the cellular endosperm has been formed. Okay, so this is one type of nucleus is discussed here, that is called as nuclear endosperm. But there are certain endosperm in which the nuclear division, as the nucleus divide, there is cytoplasmic division also, and the cell wall is laid. Means one nucleus divide, there is a karyokinesis, and after that there is a cytokinesis. So cell is formed. So two cells. Now the two cells again divide, it forms four cells. Now here I am talking about cells, not the nuclei. Okay, so such type of division when takes place, the nuclear division and the cellular division simultaneously, then that type of endosperm is called as cellular endosperm. That is called as cellular endosperm, second type. Sorry. Now the third type is the halobial one. In halobial one, at some places you see the cellular endosperm, at some places you see the nuclear endosperm in patches, So that why it is called as halobial endosperm. Halobial endosperm. Okay. So here, according to the NCERT, you have to study about the nuclear endosperm, free nuclear endosperm, which forms cellular endosperm later on. And the example is given here is the coconut. Endosperm may either be completely consumed by the developing embryo. Now, this point is very, very important. Very important. So, try to understand. Endosperm may either be completely consumed by the developing embryo, like happens in the case of pea, groundnut, and beans. Before seed maturation, the seed, before the seed get matured, completely all the endosperm has been completely used by the developing embryo, and this happened in the case of pea plant, groundnut plant, and the beans. Okay, this is one type of development, and in some cases the endosperm may persist. Okay, it means that a part of the endosperm is consumed for the development of the embryo, and the rest part is left behind. So the left behind part is called as persist, or it may persist in the mature seed. Like we see in the case of castor and coconut, and be used up during the seed germination, and that part of endosperm will be used by the seed germination. This point is clear, everyone. I'm repeating it again. Endosperm, in the use of the endosperm is there in two different types. In the first type, all the endosperm has been consumed up during the development of the embryo, like we see in the case of pea plant, groundnut plant, and beans plant. No endosperm is left behind. Now, in the second type, a part of the endosperm is consumed for the development of the embryo in the case of castor and coconut, and the small part of the embryo is persist there, left behind, and that one is used during the process of seed germination. Then what happened in such plant during the seed germination? Because at the seed germination also they require the food material, the nourishment. So at that time, from where the nourishment comes out, from where the nourishment they get, because they have already used all the endosperm at that time. So from where they get nutrition at that time of, like these plant, pea plant, groundnut plant and beans, from where they get nourishment during the seed germination. The question arises here. We will get the answer of this question. So split open some seeds of the castor, peas, beans, groundnut, fruit of coconut, and look for the endosperm in each case. Tapetum? No. Tapetum is not here. Tapetum is at, see, this is coming like, it means that you didn't understand. The tapetum layer is in the case of anther at the time of microspore development. That story is very, we left it. Okay. Now the fertilization has been taken place. The fertilization has already been taken place. There is a development of endosperm. There is a development of embryo. So there is no link of tapetum here. There is no link of tapetum. Okay. Anyways, the split open some seeds of the castor. You can take some of the seeds like castor seed you can take, peas you can take, beans you can take, groundnut, fruit of coconut, and you can split open and see where the endosperm is in each of the seed, where the endosperm lies. So find out whether the endosperm is persistent in the cereals like wheat, rice and maize. So these are the cereals. Okay, these are the cereals, monocot plant, wheat, rice and maize. Do the endosperm persist in the cereals? You have to find out that. Now we will talk about embryo because we have discussed about the persistent means the some part is left behind. Some part is left behind. That is the persistent. Okay, we will come there. We will come to that point. Embryo development at the micropylar end and the embryo sac where the zygote is situated. So which cell is involved in the formation of the embryo? The question arises here. Which cell is involved in the formation of the embryo? And that is the zygote. From where the zygote has been formed? The zygote has been formed from the syngamy process in which the egg got fertilized with the one of the male nuclei, right? And that egg is present in the embryo sac at the micropylar end. This story we have discussed in the previous class, this fertilization process, right? So embryo develops at the micropylar end, and this embryo is developing from the embryo sac only, because this embryo sac contains the zygote, and the zygote is the conversion of the egg cell. When egg cell got fertilized, it changes to zygote, and zygote ploidy is, what is the ploidy of zygote? 2n. Very good. Diploid. It is a diploid structure. 2n. Egg was haploid, right, which was present in the embryo sac, and when egg got fertilized, the zygote is formed, and that is 2n, means diploid. Most zygote divide only after certain amount of endosperm is formed. Very important line, and that is why it is highlighted. Most zygote divide only after certain amount of endosperm is formed. And why this? Because the development of the embryo requires some nutrition, and when the endosperm is formed, the nutrition is provided by that endosperm to the developing embryo. This is an adaptation to provide assured nutrition to the developing embryo. So this is the reason. Adaptation, this is the adaptation that embryo will develop later on after the endosperm has been started developing, right? And this is the assurance that the developing embryo will get the nutrition. Though the seeds differ greatly, the early stages of the embryo development, embryogeny, are similar in both monocotyledons and dicotyledons, right? Though the seeds differ greatly, there are different types of seeds. The early stages of embryo development, that is the embryogeny, and that is similar in both monocots and dicot. Okay, two types of angiospermic plants are there, monocot and dicot. I hope you all know what is the monocot and dicot. uh, by seeing the seed you can tell whether the plant is monocot or dicot. Okay, dicot means the seeds can be split into two parts, that is in the form of cotyledons, in the form of cotyledons. So dicot, two cotyledons are there. Monocot, single cotyledon. And what are the other features you see in this plant? If you talk about the root, suppose dicot plants are having the tap root system, monocot is having the fibrous root system. Now let us talk about the leaf then. Dicot will have the reticulate type of venation, whereas the monocot will have the parallel type of venation. Flower, tetramerous or pentamerous, whereas the monocot will have the trimerous. Three whorls are there only. So these are some of the differences we see in the case of monocot and dicot, but both are comes under the flowering plant, angiospermic plant. Okay, so they are telling that the early stages of the embryo development, that is the embryogeny, is similar in both the type of flowering plants, that is the monocot and dicot, right? So here we are having a figure 1.13 that depicts the stages of embryogeny in the dicotyledonous embryo. Let us see that. And these stages are the different stages, that are globular stage, heart-shaped stage embryo, and mature embryo. So let us see the figure. Here, this is the figure. So this is the zygote in the embryo sac. This is the embryo sac. This figure. This figure is the embryo sac. Yesterday we have discussed these are the class of angiosperm. Yes, you can say these are the two main class of the angiosperm, main division of the angiosperm, you can say. So this zygote is the result of the fertilization process, and that is the syngamy. And this pen is pen means primary endosperm nucleus, is the result of the triploid fusion, and both are unitedly called as the double fertilization that we have discussed yesterday. Now see this zygote part, the zygote part, which is the 2n, zygote part, which is the 2n. It divided into the two cell. Here you can see one cell is quite large and one is very small. Okay, now this small cells divide and redivide, divide and redivide, and you can say it forms a such a shape. This is the neck and this is one of the cell which do not get divide, this one, but the lower cell divide and redivide, and you can see it will form a globular embryo. First stage, globular stage or globular embryo. So first stage of the embryo formation is called as the globular stage. Now see, this part, this lower down part is divide and redivide again, and you can see it forms such a shape, like a structure, heart shape structure of the embryo, and that is why this this stage is called as heart shaped embryo. Heart shaped embryo, this is the second stage of the embryo formation, and we are discussing about the dicotyledonous embryo. To be very precise, this is about the dicotyledonous embryo. So globular stage, then comes the heart shaped stage, and then you can see the mature embryo. You can see it is having the two cotyledons here. Just just below the cotyledons here, you can see the plumule. Okay, and this embryonal axis at this particular point, you can see the radicle. So what the radicle forms and what the plumule forms at the time of germination? Can anyone can tell? Radical. What changes? What changes to radical? What, which part give rise the radical and which part give rise the plumule? Okay, I got the answer. The plumule. Plumule give rise the shoot and radical give rise the root. And that is the whole part of the plant. That is the whole plant. Shoot system and the root system. That we see. That part only we see in the plant, right? So this embryo is actually having the whole baby plant in it. Very good. Plumule give rise to the shoot and radical give rise to the root. This you have studied in your lower classes also. That's why I asked this question. Very good. Okay. So this embryo is having the whole baby plant in it. And this is present where you can see this zygote, this one cell forms such a multicellular structure, embryo. And for this development, the changing one stages to another, another to the next one, they require a lot of energy, and that energy is provided by the endosperm. Now you can understand why the formation of endosperm is very, very important. Okay. So the figure depicts the embryogeny in a dicotyledonous embryo. The zygote gave rise to the proembryo and subsequently the globular, heart-shaped and the mature embryo. Okay. So the first of all the proembryo and then comes the globular, heart-shaped and the mature embryo. These are the stages. A typical dicotyledonous embryo consists of an embryonal axis and two cotyledons. No, the proembryo is again the first part I discussed with you is the globular stage, globular embryo. Okay, the globular embryo. But before globular embryo also there is a stage in which you just see the number of cells, but you do not have any shape of that embryo. The shape comes for the globular, the shape comes for the heart shape, and the shape comes for the embryo mature one. These stages are having a proper shape, and that's why you give a particular name to it. But before that stage, there is also one stage in which the shape is not very well defined, and that is why that particular stage is called as proembryo. Proembryo. Okay. Here the embryo is having a proper stages. They reach to a definite like you can say a stage in which you can say that this is this part. You can see the cotyledon formation is taking place. You can see the radicle is there. You can see that the plumule is there, right? So those stages, the globular, the heart shape and the mature one, they are having these things and you can see that. But before that stage also, there is a multicellular stage of the embryo, but not very well defined, and that stage is called as the proembryo. Okay. So a typical dicotyledonous embryo consists of an embryonal axis and the two cotyledons. What is this? Let us see that. So here you can see this is the embryonal axis from plumule to the this root cap. This is the embryonal axis. And what is left behind here? These two cotyledons. So this is a typical embryo when it got a mature stage. When it reaches to the mature stage, you can see one axis one axis of the embryo, and that is called as embryonal axis, and the rest two things, that is the two cotyledons is there. So this is a typical dicotyledonous embryo. The portion of the embryo axis above the level of cotyledon is called as the epicotyle, which terminates with the plumule. Which terminates with the plumule or the stem tip, right? So above the level of cotyledon, that is called as epicotyle region, and that terminates where? With the plumule. The cylindrical portion below the level of cotyledon is the hypocotyle, and that terminates at its lower end in the radicle or the root tip. And root tip is covered with the root cap. Why the root? Can you show the epicotyle in the diagram? Okay. So plumule, this is the hypocotyle. This is the hypocotyle. Now here it is written, the portion of the embryonal axis above the level of the cotyledon, above the level of the cotyledon in the epicotyle, which terminates with the plumule. So suppose this is the plumule or a stem tip. Okay. So in between these two cotyledon and plumule, in between these two cotyledon and plumule region, there is a epicotyle. There is a epicotyle because though no space is there, just a very small space in between the cotyledon and the plumule. Okay. So that is the epicotyle. And here, this particular from plumule to this radicle, that is the hypocotyle. Right. So this is the epicotyle and hypocotyle region. And this root cap, why this radicle is having the root cap? To provide protection to the radicle, because they have to reach, because they have to penetrate the soil and reach deep into the soil. So do they have any function, epicotyle and hypocotyle? Yes, obviously they have some function. They are the, suppose if this plumule is not having such a tissue and it is surrounded by some of the tissues, then plumule will get the protection. The plumule will get the protection. So plumule, if suppose plumule is there and at the very next cell will be the cotyledon. Very next cell will, suppose these are the two cotyledon and this cell is just the plumule. So what can be there? So for providing protection to this plumule, some of the cell is surrounding there. Some of the cell is surrounding there, and that is the epicotyle. Similarly, the region between these two, hypocotyle. Why the hypocotyle is so large, whereas the epicotyle is very small? Because this radicle, this plumule region and the radical region has to be kept at one particular distance. They should be kept at a distance because one has to move downward and one has to move upward, right? So that is why embryos of the monocotyledons possesses only one cotyledon. Now this is the difference between the monocot and dicot. The dicot is having two cotyledon and monocot is having. So epicotyle extends, which allows the embryonic shoot to break through the soil. Okay. The epicotyle, when extend, it breaks the embryonic shoot part and allow the plumules to go upward. So it is also helping the plumule region to go upward at the time of seed germination. So this is one of the function of again epicotyle and also providing protection to the plumule region, right? Embryos of the monocotyledon possesses only one cotyledon. They are having only one cotyledon, and that is why it got its name monocotyledons. In the grass family, the cotyledon is called scutellum. Very, very important points. scutellum, and that is situated toward one side lateral of the embryonal axis. So here you can see the scutellum. This is the embryonal axis. This is the embryonal axis in the monocot. And this scutellum is present at one side of the embryonal axis, scutellum. This is the cotyledon part. This is the cotyledon part, right? And what you see in the embryonal axis of the monocot plant, sorry, you can see a shoot apex is there. Shoot apex, a part of the tissue that forms the shoot system. So that is the shoot apex. And this shoot apex is covered by the sheath-like covering, and that is called as the coleoptile. So the coleoptile is providing protection to the shoot apex. It is providing protection to the shoot apex. Okay. Similarly, when you see the lower part of this embryonal axis, you can see the radical part. The radical part. And the radical part is having the root cap also. And this whole part is again covered by the sheath-like structure. Sheath-like structure. And that is called as coleorhiza. Coleorhiza. So this is the structure of the embryonal axis in the case of monocot. So here you can see one more part, epiblast. Epiblast. What is the role of epiblast? So they are concerned for the formation of somatic tissue of the embryo proper, somatic tissue. What is the function of coleorhiza? Coleorhiza provide protection to the developing radicle. Okay, it is a sheath like covering which protect the developing, because these parts are very delicate. The shoot apex is a very delicate part. The radicle is again very delicate part. So they are covered by a sheath like covering. Okay, sheath like covering, and the one sheath like covering that protect the shoot apex, it called as coleorhiza. And similarly, the radicle is covered by the sheath like covering that is called as coleorhiza, and that also provide protection to the developing radicle. Scutellum, that is function of the doing the function of the cotyledon. Then one part is left behind, that is the epiblast. Now this epiblast is giving rise the germ cells and the somatic tissue of the embryo proper. Okay, they are concerned for the developing the cells of the embryo proper. Okay, they divide and form the cells of the embryo proper, because these cells divide and add to this embryonal axis continuously, and that is going to form the shoot apex and the root apex, right? Scutellum, that is the cotyledon. So all the parts we have discussed in the case of a monocot plant. Now this is the embryo of a typical grass plant. Grass comes under the monocot only. They comes under the monocot only. In the grass family, the cotyledon is a scutellum that is situated toward the one side, the lateral of the embryo axis. This we have seen in the diagram also. At its lower end, the embryonal axis has a radicle and the root cap enclosed in an undifferentiated sheath. It is not very much differentiated, but it is undifferentiated sheath, and that is called as coleorhiza. The portion of the embryonal axis above the level of attachment of the scutellum is the epicotyle. So we have discussed about the epicotyle. Epicotyle has a shoot apex and a few leaf primordia enclosed in a hollow foliar structure, the coleoptile. Right? So they are helping in the formation of this coleoptile structure. Right? They give rise a shoot apex and a few leaf primordia. Leaf primordia means the cells that forms the leaf primordia, and after germination this leaf primordia will forms the actual leaf. So these leaf primordia are formed by which structure? Epicotyle. Hmm? So this epicotyle. Okay. So this is a very, very important. These points are very important. Soak a few seeds in water. Now there is an activity that you have to take some seeds, okay, and you have to soak in water. Say wheat, maize, peas, chickpeas, groundnut over a night. You soak them over a night and then split the seed and observe the various parts of the embryo and the seed. You can do at your home. You can see how the embryo look like, how the seeds look like when it swells up. Which part comes out early? The shoot part, that is the plumule one, or the radical one? Which part comes out earlier? The radical. First the radical comes out, it gets established, and then slowly and slowly the plumule rises. So now we will summarize the chapter. Post fertilization, the primary endosperm nucleus pen formed by the fusion of two polar nuclei and one male gamete develops into the endosperm through the free nuclear division. So this is how the endosperm has been formed. The primary endosperm nucleus that is formed by the fusion of two polar nuclei and one male gamete, and this fusion is called as the triple fusion. The triple fusion gives rise the endosperm nucleus. Primary endosperm nucleus. Okay. The endosperm can be nuclear, cellular, and halobial type based on its developmental pattern. We have discussed, it is not mentioned in the NCRT, but still we have mentioned it. The nuclear type, the cellular type, and halobial type. I just summarize here the nuclear division, the nuclear type of endosperm. There is a continuous division of the nucleus, karyokinesis only, no cytokinesis. And once a number of nucleus are formed, then slowly and slowly there is a cell wall will be laid and the formation of cells takes place, right? Cellular type, the nuclear division and the cellular division occur simultaneously, so that is why it is called as cellular endosperm. Halobial type, a very different one, and thus in this type of endosperm, some part is having the nuclear type of endosperm and some part is cellular type of endosperm, right? So it is called as halobial type of endosperm. It serves as a nutritive tissue for the developing embryo. It serve means endosperm serve as a nutritive tissue for the developing embryo, storing proteins, lipids, and carbohydrates that is needed by the developing embryo later on. In some plants, it's completely consumed during the embryo development, while in others, like cereals, it persists in the mature seeds. It persists in the mature seed. Endosperm is there. Some part of the endosperm is left. But in some seed, like peas, beans, all the endosperm is consumed during the embryo formation. So where the food material, this question I have asked, from where the food material they get at the time of germination? From the cotyledon. Cotyledon also stores some sort of food material, and that food material is given at the time of the seed germination. Okay. The endosperm is typically triploid and plays a crucial role in the seed development and early seedling growth. So this is all about today's lecture, but we will have our DPP. DPP. So here is our DPP, today's DPP, and the first question is here. The first question is here. Which of the following is true about the endosperm development? So there are some statements regarding the endosperm development. The first statement, it occur after the embryo development. It occur after the embryo development. Second point, the primary endosperm cell is diploid. The primary endosperm cell is diploid. The primary endosperm cell is diploid. Now I hope the screen is visible. Okay, thank you. Third statement, free nuclear endosperm is a stage where the cell walls form immediately. And option 4 is coconut water is an example of free nuclear endosperm. So if we see the first statement, it occurs after the embryo development. Mostly it occur before the embryo development. So the first statement is incorrect. The primary endosperm cell is diploid? No, it is triploid. So the second one is also incorrect. Pre-nuclear endosperm is a stage where the cell wall forms immediately. No, the cell wall forms very late. It does not form immediately. So this statement is also incorrect. But see the fourth one. Coconut water is an example of the free nuclear endosperm. Yes, this is the correct statement. So you also, all of you have opted the D statement only. Very good. We'll save and move to the next question. In a typical dicot embryo, the portion of the embryonal axis above the cotyledon, very important. A typical dicot embryo, the portion of the embryonal axis above the cotyledon is called hypocotyle, B epicotyle, C radical, and D coleoptile. So you all know what is coleoptile, you all know what is radical. The confusion is between the A and B, hypocotyle and epicotyle. Okay, the portion of the embryonal axis just below the cotyledon is called as hypocotyle, and the portion of the embryonal axis above the cotyledon is called as the epicotyle. So you all have given the option B as the correct one. So I'll opt it and move to our next question. Which seeds are categorized as albuminous? Albuminous seeds. A. Pea and groundnut. B. Castor and maize. C. Beans and peas. And D. Groundnut and beans. Albuminous and ex-albuminous. Two seeds are there. So, pea is an ex-albuminous, but groundnut is albuminous. Castor and maize are the albuminous. Beans and peas both are ex-albuminous. Groundnut and beans, so beans is again the ex-albuminous, but the groundnut is albuminous. So the one option, that is the castor and maize, they both are albuminous. So we will opt this as a correct answer. We'll save and move to our next question. What is the primary function of the micropyle in a seed? Function of a micropyle in a seed. Let us see the option. A. Storage of nutrients. B. Protection against the pathogen. Facilitation of oxygen and water entry, and attachment of the ovary wall. The first option, storage of nutrients. This function is of endosperm, not the micropyle. So the option A is incorrect. Protection against the pathogen. Seed coat, a hard seed coat is there that provide protection to the seed, and do not allow any pathogen to get enter inside the seed, right? So this is again not the function of the micropyle. Coming to the option C, facilitation of oxygen and water entry. That micropyle, the small pore is there through which the oxygen could enter inside the seed, and also the water, the entry of water during the process of germination, that also occur through this particular micropyle only, right? So option 3 is the correct. Attachment of the ovary wall. Micropyle is not there for the attachment with the ovary wall. So that is by the funicle, and the placenta is there. So the correct option is option C, facilitation of oxygen and water entry. That is the function of the micropyle in a seed. So we will save and move to our last question. Which of the following is an example of false fruit? Okay, funicle is the stalk-like structure. Funicle is the stalk-like structure, and this stalk-like structure with the help of placenta is attached to the ovary wall. Okay. Which of the following is an example of a false fruit? False fruit. Mango, true fruit. Ovary forms the mango. Guava, true fruits from the ovary. Groundnut, true fruit from the ovary. But the apple is formed from the thalamus. Ovary is embedded inside it, but what is the edible part? The food is stored in the thalamus one, right? So the apple is having the false fruit. We will save it and submit the test and see the results. So our all the answers are correct. In fact, your all answers are correct. So this is all about today's session. Okay. We will have 2 more sessions to complete this particular chapter. And then we will see this whole chapter with an eye of, with the eye with the, for the NEET examination. 2 more lecture will be there, right? So there we will see this whole chapter in a very great detail. So this time we are discussing about the NCERT. Okay, so thank you very much for attending today's lecture.